When you think about the global pandemic, it really feels like this massive planet-wide event. But at its core, it's a conflict happening on a microscopic scale. And it all boils down to one tiny, absolutely critical piece of the coronavirus, the receptor binding domain, or RBD. This little part of the virus's spike protein is basically the master key. It dictates everything: how the virus gets into our cells, how our bodies fight back, and even how we design the vaccines to stop it. If you want to understand this virus, you have to understand the RBD. So let's break it down. All right, so here's what we're going to cover. First, we'll look at the RBD as the virus's master key. Then, we'll get into the nitty-gritty of how it actually gets inside our cells. After that, we'll explore our immune system's really complex response. Then, we'll get into the cool science behind designing smarter vaccines. And finally, we'll touch on the surprising role our own genetics play in this whole battle. So, first up, the viral master key. It's so important to think of the RBD not just as some random part of the virus. It's a highly specialized tool that has evolved for one purpose: to pick the lock on our cells. So picture the coronavirus. It's covered in these things that stick out called spike proteins. And right at the very tip of each spike is this crucial component called the receptor binding domain, or RBD. Its one job and one job only is to find and latch onto a receptor on the surface of one of our cells. It's that very first point of contact, the handshake that kicks off the entire infection. Now, this is where it gets really interesting. The whole process is incredibly specific. Think of it like a lock and a key. The SARS-CoV-2 virus has a very particular key, its RBD, and it's shaped perfectly to fit the ACE2 receptor lock, which is found on lots of our cells. But its cousin, MERS-CoV, has a totally different key that fits a completely different lock, the DPP4 receptor. This specificity has a name. It's called viral tropism, and it's what determines which cells, which tissues, and even which animals a virus can infect. It's everything. Okay, so we've got the key, but how does it actually turn the lock? This next part is all about the physics and the chemistry of that invasion, the actual mechanics of what's going on. The RBD isn't just sitting there waiting around. It's actually in constant motion, flipping between two main positions. In the down state, it's kind of closed up, hiding its most important parts from our immune system. But to actually infect a cell, it has to flip up into the up, or open position. This move exposes the sites it needs to attach to our ACE2 receptor. But, and this is a big but, it also makes the virus vulnerable to our antibodies. It's a huge gamble for the virus, every single time. So, you might be wondering, what made SARS-CoV-2 so ridiculously good at infecting humans? Well, a huge part of the answer comes down to something called binding affinity, which is basically just a fancy way of saying how sticky it is. Compared to a close relative, a bat coronavirus called RATG13, the SARS-CoV-2 spike protein evolved to have a much, much tighter grip on our human ACE2 receptors. And just how much tighter are we talking? About 1,000 times tighter. No, that's not a typo. 1,000 times. This isn't some small, gradual improvement. This is a massive evolutionary jump in how well it can grab onto our cells. And that translates directly to how infectious it is. Okay, so how on earth does it get that incredible grip? It all comes down to tiny little changes at the molecular level. You can see here that the SARS-CoV-2 RBD forms these crucial electrostatic bonds, called salt bridges, with our ACE2 receptor that its bat relative just can't make. And on top of that, one specific amino acid, Phe486, slots perfectly into a little pocket on a receptor, acting like a molecular anchor. It's this combination that creates that super tight, high-affinity bond. And believe it or not, the RBD isn't a one-trick pony. It has other tools in its belt besides just grabbing ACE2. It has this unique sequence called an RGD motif that lets it bind to other things on our cells called integrins, and that might be linked to some of the systemic problems we see, like blood clots and inflammation. It also has these 4 strange inserts that look a lot like proteins from HIV-1, which might help it bind even better and infect an even wider range of cells. Right, that brings us to the other side of this fight, our own immune system. The RBD is the #1 target for our defenses, but as you're about to see, that response can be a real double-edged sword. So let's start with the good news. Because the RBD is absolutely essential for the virus to get in, it's the perfect target for our immune system. If we can make antibodies that just block the RBD, we can stop the virus dead in its tracks before it ever gets inside a cell. This is what makes it such an ideal candidate for a vaccine. So, if it's such a perfect target, what's the catch? Why isn't this super simple? Why can't we just show our immune system the RBD and call it a day? Well, here's the problem. When we make certain kinds of vaccines, we have to pull the RBD off the virus and isolate it. But that artificial process accidentally exposes parts of the RBD, these little sites called epitopes, that are normally hidden when the virus is whole. And some of these newly exposed sites can act like decoys, basically tricking and distracting our immune system. And you can see exactly what happens because of this decoy effect right here. On the left, you have the ideal immune response. It focuses all its firepower on the real target, blocking the virus. Perfect. But on the right, you have a distracted response. It's wasting its energy making useless antibodies that attack the decoys. This means the real virus can just slip right past, which makes the vaccine less effective. And if that wasn't enough, there's another complication called molecular mimicry. This is basically a case of mistaken identity. It happens when a piece of the virus looks almost identical to a piece of our own human proteins. As you can see, the spike protein has a short sequence that's also found in our own bodies. The danger here is that our immune system, in trying to attack the virus, gets confused and starts attacking our own cells by mistake. And this isn't just a theoretical problem. The implications are really serious. This kind of cross-reactivity has been suggested as a possible cause for a whole range of post-COVID conditions. An immune response that's supposed to be fighting the spike protein might accidentally target human proteins like myelin in our nervous system or alpha-myosin in the heart, potentially leading to serious neurological and cardiovascular damage. So, with all these challenges, you've got decoys, you've got mimicry. How in the world do you design a better, more focused vaccine? Well, the answer lies in some really clever science, a strategy that lets us focus the attack where it matters most. This strategy is called immunofocusing, and it's this really elegant three-step process. First, scientists figure out which parts are the good targets and which are the bad decoys. They basically give them a score. Second, they engineer the vaccine to make sure it prominently displays all the good parts that create protective antibodies. And third, and this is the really clever part, they use these complex sugar molecules called glycans to physically cover up or mask the bad decoy sites, making them totally invisible to the immune system. And we know this works. It's not just an idea on a whiteboard. In experiments with the MERS virus, scientists showed that an RBD designed with this immunofocusing strategy, the one with the masked decoys, produced a much, much stronger neutralizing antibody response than the regular old RBD. It's powerful proof that we can intelligently guide our immune system to be way more effective. All right, for the final piece of this incredibly complex puzzle, we have to talk about, well, us. The host, the person getting infected, is not a passive bystander in this. Our own genetics can play a huge role in how this all plays out. Remember that lock and key analogy? Well, it turns out the lock, our ACE2 receptor, isn't exactly the same in every single person. Tiny genetic variations can change the amino acid sequence right where the virus binds. And these little changes can alter the shape and the charge of the receptor, making it either easier or harder for the virus's RBD key to fit. And the data on this is just wild. Models predict that some of these AC2 variants make the bond with the spike protein way stronger. The G211R variant, for example, creates a much better attraction. What's more, this variant is most common in European populations, and these kinds of differences around the globe might help explain why we see different patterns of susceptibility in different places. So when you step back and look at the big picture, this one little domain, the RBD, is sitting right at the center of this entire biological drama. It's key to the virus's strategy, it interacts with our own genetic susceptibility, it drives the disease, and it's both the perfect target and a frustrating challenge for our immune system and for our vaccine designers. It perfectly captures this complex, ongoing arms race between a virus and its host, and it leaves us with a critical question. As the virus continues to evolve its key, how quickly can we redesign ours?